So my name is Elena and I want to thank all the organizers of this workshop for opportunity to talk about slightly different product. This is not a single trait product, but rather a product that intends is to modify such a complex qualitative trait as yield under drought stress. And just to set up stage, I just want to give you a little bit context about effect of the drought stress on the maize yields. And effects are usually very dramatic, and it depends when drought will happen. If drought happens during flowering stress, then effects really severe. And if it's during grain filling stress, then effects may be moderate. But still, in all cases, when drought happened, the economical effect is significant. Yield drops. 2012 was a year when overall yields dropped below 150 bushels per acre. And what you see here in this field, that one of the major effects that drought has on corn plant or maize plant is the growth. Plants stop growing. And I was really surprised to see plants in the field in 2012 that looked like a pineapple. Naturally, there was no nodes elongations. So when we're talking about qualitative product as that changes yield, really this development of such product is somewhat reminds the breeding process. And the reason why, because we're looking for transgenes that actually will interact with endogenous pathways in the plant. So we're not looking for something new to be created, we're actually looking for genes that will interact and modify response of plant to the specific conditions. So breeders did it very successfully, and over years they increased yield, maize grain yield significantly, starting from late 1930s. Some of that was driven by agronomy practices, but large part was driven by genetics. And that means that during the breeding process, breeders actually created completely new gene combinations that were responsible for a variety of observable phenotypes. And yet, breeders had no idea what genes or pathways were involved in those changes. So as breeders initially were not really breeding for drought, but they were doing selection year after year, and each year brings completely different environment. And it's interesting that when a lot of different hybrids that were produced in different years, starting in 1930 and up to 2010, were all tested together in the same field in 2009, 10, and 11. What we saw that newest hybrids have really significantly improved tolerance to stresses because they have significantly improved water use efficiency. It means they produce more grain per inch of water used. So those changes in water use efficiency and drought tolerance actually, as we know as a crop physiologist, plant physiologist, usually associated with very significant changes on the whole plant basis. It's usually you need to have better resource capture and assimilation. You also will see changes in the growth habits, developmental habits of the plant. And those changes happened when a lot of different networks and pathways being affected. So this is just a few examples, simple examples that I want to show that usually associated with newest hybrids. What you see here, one of the interesting that leaf angle change that allows plant to have upright leaves and compared to leaves that were flat in older hybrids, but that allows plant to capture more photosynthesis and increases photosynthetic area when we plant them at higher density. And it's an interesting example because this particular change is really caused by one specific gene. And using biotechnology approach, we can easily reproduce that change. We can put the same gene and we will get exactly the same phenotype. So there are other changes that are more complex. For example, growth rate. Growth rate is very complex trait, and usually it's governed by many different genes. But what we saw also that newest hybrid here are always show very increased growth rate during late vegetative stages compared to the older hybrid, and it also leads to increased ear growth rate, which correlates directly. And what that allows is to establish larger sink potential in newest hybrids. Senescence is another example. the new hybrids show delayed senescence, and it allows to extend the reproductive grain filling period. But senescence also is a very complex trait, and it's interactive, and it's usually correlates with many changes on the roots level. So therefore, when breeders increased yield, they change many different phenotypes, they change many different pathways. Some of those phenotypes we now evaluating, a lot of them we still don't know, and we definitely don't know genes that involved into all these changes. So another example, when we actually did the experiment, when we took a lot of different hybrids, and it's approximately 500 different hybrids, and we put them in an automated greenhouse where we can actually control environments, so we can eliminate environmental aspect. And we saw how they react, and the growth rate reacts to all different, controlled conditions. And when you look at the normal condition, you see that it's very broad distribution of the growth response, and it's not really one population. When we shifted to water stress and nitrogen stress conditions, that distribution completely changed. Hybrids react completely different to these conditions, and it's driven purely by genetics, because environment here is well controlled. Unlike breeding lines, when we look at 2,500 of transgenic events that originate from different genes, what we saw that the population was very tight, the distribution is very tight, and really there is all this variety of different transgenes has no ability to shift significantly the response of this population. So metabolites is another story, as many speakers already mentioned that. When we did analysis on several metabolites across approximately 200 different hybrids, what we see, this is all normalized data just to be able to put it on one graph, but what we saw that variability in metabolites across different hybrids is very, very broad. It's several fold differences, and we never actually evaluated any risk safety of changes in those metabolites. When we looked at the one single line and we saw what factors will drive the changes in metabolites, then development was the major factor. And then, of course, environmental conditions changed a lot response of those hybrids. So therefore, even though the breeders were consistently breeding for yield, they created hybrids with different combinations of the genes that drives different combinations of the observable phenotypes, and that variability is very broad. All right, so I said at the beginning that creating the qualitative trait that improves yield is somewhat reminds us the breeding process, and I just wanted to put it together here. So when breeders start, they combine different genetics, multiple genes, and in biotech approach, we usually try to work with one, maximum two genes. So then breeders test many lines for desirable phenotypes, and that's yield. Well, that is very similar because we create and using biotech approach many different events, and we are testing them for desirable and undesirable effects. And just like in the breeding process, we eliminate undesirable effects in the selection process. So as a result of that, we select in 1 or 3 events for commercialization. And of course, as all previous speakers mentioned, there is a lot of work goes into ensuring that we have good insertion, we know the insertion site, we have knowledge of the flanking region, etc. So all that included. And once we have an event that we believe is a clean event that produces the yield that we want, we put it in through very thorough safety evaluation based on the regulation that we have right now. And part of that right now for traits like qualitative traits is evaluation of gene mode of action. None of that, of course, happening with the breeding lines, but at the end of the process, breeders put forward several lines for commercial release, and after regulatory approvals, we have ability to commercialize event as well. The outcome of this is that as a result of breeding, we have so-called native traits that produce better yield. However, we know much less, they characterize much less, and the function not clear and not specified. When we're talking about biotech traits, they are usually well characterized and they're targeted at specific function, and that's what I would try to show you today. I will talk today about our first drought product. It's we call it DroughtGard. And this is really a combination of the breeding germplasm. I think we're at the point right now when we're trying to utilize the advances that breeding made, developing the best breeding line, and then layer it with biotech approach where we can lift the performance of this best hybrids even more. And that's exactly the case here, where we used our drought-resistant germplasm and we added drought-tolerance gene. So what is the gene? Gene is, we call it CSPB, it's cold shock protein, cold shock domain protein, and it's a small protein. It is an RNA chaperone, and the main function is for this protein is to bind to double-stranded RNA, meld the double-stranded structures, and allow RNA to be translatable during environmental stresses. And it is from Bacillus subtilis, and it allows this very nice microbe to actually survive unfavorable conditions and continue growth when conditions become favorable. So the good part about up front we knew that this protein should be very safe because the protein has significant history of safe use. The bacteria itself used in Japan to ferment natto, and natto is very popular food in Japan, makes their way here to United States, probably Canada as well, and it's also part of fermenting a lot of different foods in Asia, including cabbage, carrots, etc. We actually measured the presence of this protein in the food, and we know that exposure to this protein is quite significant because protein is easily detectable in all those foods. So protein was originally tested in model plants and Arabidopsis, then it was tested in rice. We saw the effect, put it in maize, and then the question was how we will evaluate what this protein actually does in maize and how we will prove that it does something that we intended to do. So our hypothesis was to start with that this protein will work as intended, as RNA chaperone, and therefore it will protect growth of the transformed maize plants during stress. So the rest of my talk would be about how we actually try to show that it's exactly what this protein is doing. And of course, by doing so, we had large chance to either prove or disprove this hypothesis. So we started with molecular mode of action. There's a lot of data were produced. What I want to show you here, only two pieces of data. One that clearly shows that protein binds only to RNA and it does not bind DNA. And that was big because, again, when we're thinking about unintended effects, it's the question if it binds to unintended structures, it may have potential to do something that we didn't want that protein to do. In this case, we saw that no, the protein binds only to RNA as we expected. Moreover, when we looked at what type of RNA it binds, what we saw here that we found this protein only in polysomal fraction. And that means that protein binds only to actively translatable RNA and trying to protect that RNA. It's not binds to free RNA. So, so far at molecular level, the protein showed that it acts exactly as it does, and activities actually show that it does melt double-stranded RNA structure and continue to support RNA translation. So it was very nice. This work was done several years ago, but it was very nice to see that last year the paper came out about endogenous Arabidopsis gene that homologous to CSPB, and actually it was shown that it acts exactly the same way in Arabidopsis. So that was really nice. All right, so molecular level is one story, but then we really needed to show, or at least to prove ourselves, that at the physiology phenotypical level, we do see effects as we expected. And of course, we started with growth chamber experiments. It's the first line of experiments that we will do. And what we saw is sensitive. So what we saw first, that when we applied drought, and this is days after we applied drought to growth chamber grown plants, first we saw that actually the pot weight, when we weighted the whole pot, was increased in our transgenic plants compared to the control, which is a little bit different than we thought it will happen. But Then when we looked at the shoot fresh weight, what we saw that plants actually were significantly smaller. But because they were smaller, they also used less water. Therefore, the whole system weight was larger. So what we concluded from that there is a growth response to drought stress, immediate response. So the next, we needed to understand if the response, the way we see it, how we get in yield. Because if plants are smaller and simply smaller, usually it's not translatable to increase yield at the end of the season. So what we started looking at in more details, how this gene affects leaf elongation or leaf biomass growth. And what we saw that, again, the same story. When we apply drought, in this case, drought was applied in greenhouse around flowering, and when we apply the drought stress, very quickly, the transgenic plant, it's here, showed immediate decrease in the growth response. So it stopped growing much faster than control plant. But after several days, when control plant growth rate went down, and unfortunately you don't see here more data points, the transgenic plant continued to grow with the same rate. And that's where change happened. So it shows very rapid decrease in the growth rate, but after that, it's continued to grow with the same rate when wild type actually growth rate went continuously down. And what happened as a result of this change was that, remember that graph I showed you at the beginning, that plant growth rate during flowering really relates directly with ear development rate. And it's exactly what we observed here. So after several days, after 5 days of drought stress, ear development was at the same stage. Well, size of the ear was the same. But after prolonged drought stress, transgenic ear development was increased compared to the control plant. And what we ended up at the end of the stress as a larger ear developed on the transgenic plants. So the question was that, okay, it's a growth chamber experience. We see what we expected to see. And I should notice that on previous slide that we also obviously did the same experiments under well-irrigated conditions, and there are no differences in the growth rate of the leaf or shoot or the ear when no drought is applied. And also we didn't see any other changes, at least observable in the greenhouse at that point. But the main question, what does it do in the field? And that's where we thought how we will evaluate that in the field. And the question with the field that it's very hard to measure growth rate in the field, that measurement is not precise and it's very resource consuming and it really doesn't give you good data. How to get good data to answer the question? So what we decided that if our hypothesis is correct and plant decrease the growth rate, then they will use less water because that's what the smaller plant will do. And there is a way to measure it with high precision. You can measure it using sap flow sensors, so it attached to the plant itself and directly measure how much water plant can pull. You also can measure soil moisture using capacitance probes, and we did both. So when we used both approaches, we saw exactly the same result. And here's an example I'm showing you comes from the sap flow data. So when we apply drought, now in the field, again, the drought was applied around flowering stage, but the timing that during which the growth rate was reduced was significantly longer, obviously, because we're now dealing with soil capacity to hold water and for drought stress need to develop over time. But it was interesting that we saw again a very rapid response very rapid response, and the transgenic plants were consuming significantly less water in this case than control plant under drought conditions. And there's no differences that we can detect was noticed under normal irrigated conditions, and we also did test under rain-fed fields where we had plenty of rain, and again we didn't see any difference whatsoever. So again, this data confirmed that the first response is very rapid, and most likely plants continue to grow at better rate later on as adaptation to stress due to the presence of the CSPB protein. So that data and many other data, of course, allowed us to build the hypothesis how the gene works, what does plant do. And what we concluded that under normal conditions, we wouldn't see any difference. Plant would be exactly the same, it will grow with the same rate, but when drought happens, we will see very rapid, more rapid initial growth reduction in the transgenic plant. So this is control. So obviously this changes relative to this control line. There is some savings in water during that period. It's not very significant, but most likely it will help plant later on if there's no rain. But in general, the growth rate itself is a mode of action of CSPB protein. So it's a rapid decline and then adaptation to the stress, and it goes and continues to grow with a rate higher than wild type, eventually leading to the increased yield at the end of the season. Now, I just want to say that there are a lot of different things you can measure and a lot of different things you may miss. And if we wouldn't have original hypothesis of what we believe the gene is doing, we probably would never do the measurements of that kind. So we probably would miss on all major mode of action aspects of this gene and won't be able to describe it. So is it important to describe mode of action? We believe so, because in this case, all what we're trying to show that the gene binds to the same molecule on molecular level. It shows us intended effect, and it doesn't show any other effects. And we did, of course, took all agronomical measurements as we required to, and we saw no changes. We added photosynthesis, no changes we saw under well-watered conditions and under drought stress. The main difference, again, it's developing ear, which correlates to the growth rates, obviously yield components, and yield itself. So we'll obviously looked at metabolites. The gene, based on our hypothesis, is not metabolic gene. We wouldn't expect to see any changes. And as expected, we didn't. As I showed you before, the drought stress itself impacts metabolites significant way, developmental stages as well. So you could see here that both transgenic and control plants respond very similarly to developmental stages. It's also responds similarly to the drought stress. However, there is no any significant or meaningful differences that you can see between transgenic and control. So in this, I just want to quickly get back to that slide that I showed in the beginning, and just to emphasize that when we looked at, again, it's around 2,500 different transgenic events, what we saw that it doesn't matter what genes that those events were a product of. In majority of the cases, we saw almost no variability in the metabolites, at least meaningful variability. And the context of variability we see between different hybrids, between different developmental stages, between different environments. The changes that we see due to the transgene, I would say, well, it's not meaningful, at least not biologically meaningful. All right, so what are the main thoughts here? As you notice, I didn't talk today about safety assessment at all, more about mode of action. But we do believe that understanding of mode of action and having hypotheses about mode of action and actually proving or disproving this hypothesis should help us and you as regulators to understand what is the potential risk and what the potential absence of risk for products like that. Because I remember at the beginning somebody said that for products like BT and herbicides resistance, we know everything. It's new products that would be different. This is different product, but it seems that if we have ability to approach it on a rational basis from hypothesis building, and therefore build the safety assessment based on that. So just as what my final thoughts, that breeding does introduce novel gene combination with very, very broad effects. We don't know those effects. We haven't evaluated all these effects. We only scratched the surface at this point. The conventional germplasm lines show variability in phenotypes, responses, growth responses to abiotic stresses, and they actually do represent major source of variability. So biotech, when we're thinking about qualitative traits like yield, doesn't introduce genes that's supposed to interact with endogenous pathways. So they have to change plant in some way so that plant responds better to the stress. So therefore, it's not equivalent from that perspective. But the effect should be intended and could be detectable. And other effects on plant phenotypes, physiology, metabolites can be easily assessed when you base that on the hypothesis. I also want to emphasize that in this case, breeding and biotech products, they go through similar process of selection for unintended characteristics. And just like in the breeding, biotech has great ability to eliminate all unintended or undesirable effects during event selection process. And just as a summary of those slides with all different metabolites, etc., it is very unlikely that biotech product will represent a major source. Actually, they do not represent a major source of unintended effects when we compare it to the variability that already exists due to the breeding. Thank you. That's all what I have today.